Krebs cycle. And how we're going to do that is by drawing. So here I draw a line and our circle because it's a cycle. In our corner here, I'm going to draw or write down our key. And I remember Cs, Fu, Mal, and O. That's our key. First off, we have pyruvate, and a pyruvate comes from the glycolysis process. So we have pyruvate, that's our 3-carbon, and then acetyl-CoA, that's our 2-carbon substrate. Then we're going to use our key, C-I-A-S-S, Fu, Mal, and O. So we used our key. Now we just got to fill it in. Citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate, and that's all of our substrates that's involved in the Krebs cycle. So what I advise is that you stop the video, rewind, replay this, and get the core down. Get it down, because when you remember the enzymes, you'll get the names of the enzymes from the substrates. So you get this down, you can get everything else. So stop the video, replay, and practice, practice, practice. After that, come back and we'll do the enzymes. Next up, we have our enzymes. We're going to remember those. So most of them are dehydrogenases. So we're going to we're going to leave that for last and focus on synthase. Now the 2 synthase that are in the Krebs cycle are between succinyl-CoA and succinate. So we're going to write synthase here and then leave the name blank. And then our other synthase is here between oxaloacetate and citrate. So we're going to leave a blank for the name and then synthase. Our next enzyme is a conitase. And a conitase is between citrate and isocitrate. And then our next enzyme is fumarase. And that's between fumarate and malate. So remember all of these. The rest of the enzymes are dehydrogenases. So we have a dehydrogenase here. Leave the name blank. A dehydrogenase here. Leave the name blank. No, get the picture? Dehydrogenase. 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 All right, on to the names. So the first one's between pyruvate and acetyl-CoA. Bring over the name. So it's pyruvate dehydrogenase complex. So all you have to do is just bring over the name. Same thing with our next enzyme, dehydrogenase enzyme. Bring down the name. Isocitrate dehydrogenase is between isocitrate and alpha-ketoglutarate. Bring down the name. Between alpha-ketoglutarate and succinyl-CoA, what? Bring down the name. Alpha-ketoglutarate. With the next dehydrogenase, what are you going to do? Bring over the name. Succinate dehydrogenase, and it's between succinate and fumarate. Our next dehydrogenase, bring over the name, and it's malate dehydrogenase. Now for our synthases, so between citrate and oxaloacetate, what are you going to do? Bring over the name. Citrate. Citrate synthase is between oxaloacetate and citrate. Between succinyl-CoA and succinate is succinyl-CoA synthase. Bring down the name. All right, and these are all of your enzymes. So again, what you have to do is pull out the ones that are not dehydrogenases, fumarase and aconitase. Then you do the synthase, citrate synthase, and succinyl-CoA synthase. Now the rest of them are dehydrogenases. So you fill in dehydrogenase, and then you just fill in the names of the substrate previous to the enzyme. And you have your Krebs cycle with your substrates and with your enzymes. Next, we're going to go over the reactions in the Krebs cycle. Stop this video and practice this, all of your enzymes, and then go ahead and practice all of the substrates. Practice, practice, practice. All right, it's time to remember the reactions in the Krebs cycle. So between pyruvate and acetyl-CoA, we have carbon dioxide and NADH. Between isocitrate and alpha-ketoglutarate, we have NAD plus converting to NADH and carbon dioxide leaving the cycle. Same thing between alpha-ketoglutarate and succinyl-CoA, NAD plus converting to NADH and carbon dioxide leaving the cycle. Between succinyl-CoA and succinate, GTP leaves the cycle. Our next one, we're going to remember F, fumarate and succinate. Between these two, F, FAD, converting to FADH2. And our last one, between malate and oxaloacetate, NAD plus converting to NADH. All right. Carbon dioxide, NADH, between pyruvate and acetyl-CoA. Between isocitrate and succinyl-CoA, there are two reactions. NAD plus converting to NADH, and carbon dioxide leaving the reaction. Between succinyl-CoA and succinate, GTP leaves the reaction. And between succinate and fumarate, FAD converts to FADH2. And then between malate and oxaloacetate, NAD plus converts to NADH. Now remember, this is one cycle of one pyruvate. When we had glycolysis, it was resulting in 2 pyruvates. So we are going to go through this twice for both pyruvates. And this is the Krebs cycle.